Okay, imagine your body's uh, hardest working filters, just tirelessly working away day in, day out, keeping everything balanced, you know.
You mean the kidneys? Yeah, absolutely vital.
Exactly, the kidneys. They process what? Something like 180 liters of blood every single day, just removing waste, toxins.
It's an incredible workload.
But here's the tricky part, right? Chronic kidney disease, CKD.
hmm
It often just creeps up on you silently, slowly damaging everything, and you might not feel a thing until it's, well, really quite advanced.
And it affects millions. It's a huge global health issue, really impacting people's lives.
Which is why today we're doing a deep dive into something genuinely groundbreaking, a scientific discovery that offers, well, real hope, not just for earlier detection, but maybe even prevention, treatment.
Yeah, this is potentially huge for chronic kidney disease.
It's being called a world first, and it could fundamentally change how we think about kidney health. We found this great article on SciTech Daily that really sparked our interest.
And it really hits on a massive, long-standing problem. The thing is, until now, we just haven't had a reliable way to check on the health of the kidney's tiniest structures, those little
Yeah.
blood vessels.
Especially not early on.
Exactly. The damage can just build up for years, totally under the radar of the usual tests. So yeah, our mission today is to unpack this discovery. What is it? How does it work?
And what does it actually mean for patients? That's the crucial part.
For kidney patients, definitely, but maybe even beyond that, for other conditions too.
Okay, let's unpack this thing. Get ready for some truly fascinating science.
Let's do it.
So, why is CKD so hard to catch early? We know the kidneys are vital, but where's the disconnect?
Well, a lot of it comes down to those delicate kidney blood vessels you mentioned. The damage often starts there deep inside.
Like the plumbing getting corroded from the inside out, almost silently.
That's a good way to put it. And specifically, we're talking about something called peritubular capillaries. These are super important.
Peritubular capillaries. Okay, what makes them so special?
Well, they surround the kidney tubules, the bits that actually do the filtering and processing. After the initial filtering, these capillaries do two critical jobs. First, they pull back useful stuff, nutrients, water into the blood so you don't lose it.
Right, reabsorption.
Exactly. And second, they deliver oxygen and nutrients to the kidney cells themselves. They keep the kidney tissue alive and working. They're like the kidney's lifeline.
Okay, so absolutely essential.
Totally. And what happens is when there's kidney damage, maybe from surgery, cutting off blood flow temporarily, or long-term things like diabetes, high blood pressure, these tiny vessels get lost. They die off.
And when they're gone,
The kidney just can't function properly. It's this sort of downward spiral that starts at this microscopic level.
which really highlights the gap we had, right? The article mentioned there was no dependable biomarker before this discovery to check on these capillaries.
Precisely. Traditional kidney tests, like creatinine levels in the blood, they often only show a problem when overall kidney function has already dropped quite a bit.
Meaning a lot of damage might have already happened to these microvessels.
Yes, exactly. The warning comes too late. You're measuring the consequence, not the initial problem in the microvasculature.
So how is a microRNA biomarker different? How can it possibly detect this sort of hidden damage? What were the hurdles?
Well, a biomarker is just a measurable sign of what's happening biologically, right? Could be a molecule, a gene, something that tells us about health or disease.
Like a little flag going up.
Kind of, yeah. The problem with these peritubular capillaries is they're tiny and buried deep in the kidney. So standard blood tests don't really see them specifically.
You need what, a biopsy?
Traditionally, yes, a kidney biopsy could show it, but that's invasive. You can't just do biopsies on everyone for screening, especially people who are already at higher risk. It's just not practical.
So the challenge was finding something non-invasive, something in the blood, maybe.
Exactly. A specific sensitive signal in the blood that acts as an early warning for the specific type of damage. That was the mission, finding that molecular messenger.
Okay, so with this hidden damage happening and no good early warning system, the need was definitely there. And this is where the Canadian researchers at COCHUM come in. The research center linked with Université de Montreal.
Right. This is where the breakthrough happens.
They identified a specific microRNA, and this apparently could change everything.
It really could. So a microRNA, just to quickly explain, it's a tiny piece of RNA. It doesn't make proteins itself, but it acts like a dimmer switch for genes.
A regulator.
A master regulator, exactly. It binds to other RNA molecules, the blueprints for proteins, and basically fine-tunes how much protein gets made. Sometimes it even shuts production down completely.
So tiny molecule, big job.
Very big job. And these researchers found one called MIR423-5P. And what's remarkable is its ability, it seems to protect those tiny blood vessels, the peritubular capillaries we talked about.
Protect them.
It
How?
helps safeguard them and maintain kidney function even after a serious injury. It's like they found this tiny guardian molecule for the kidney's most vulnerable parts.
Wow. And we should definitely give credit where it's due. The researchers, Dr. Marie-José Hebert and Dr. Heloise Cardinal, both medical professors at Université de Montréal.
Right. They hold the Shire Chair in nephrology, renal transplantation, and regeneration, a prestigious position.
And also Francis Mignot, Dr. Hebert's research associate. Their work got published in JCI Insight.
Which is a top journal, yeah.
Yeah.
And it's important to stress, they didn't just stumble on this randomly. Their research was specifically looking at why these peritubular capillaries decline.
They suspected this was a key part of chronic kidney failure.
Exactly. They knew this microvascular damage was crucial but often overlooked. So they were already focused on finding the root cause, understanding that the big failures often start with these tiny breakdowns.
That focus clearly paid off. So how did they actually find this specific microRNA? I imagine it started, like a lot of science, with careful work in the lab, animal models.
That's right, it often starts there. Francis Mignot, the first author, he described how they initially saw something interesting in mice.
What do they see?
He said, and I'm quoting roughly here, "We first saw fluctuating levels of miR-423 5P microRNA in the blood of mice with acute kidney injuries."
Fluctuating levels.
Yeah.
So it changed when the kidneys were hurt?
Exactly, that was the aha moment. It suggested this microRNA wasn't just floating around, it was actively involved when the kidney was under stress or damaged.
That's a huge clue.
A critical clue, because then the next logical step, the really crucial one, is to see if it holds true in humans.
Right, the big question, does it work in people? And this is where it gets really relevant for you listening. It wasn't just mice.
No, they confirmed it. They looked at 51 kidney transplant recipients from the CIQ Biobank.
51 actual patients. That shows real rigor, doesn't it? It's not just theoretical.
Absolutely. And using transplant patients was smart. These patients often have that temporary blood flow cut off during surgery, which can cause acute kidney injury and damage those microvessels.
The exact scenario they were studying.
Precisely. So seeing the same miR-423 5P fluctuations in these patients give really strong evidence that this microRNA is a relevant biomarker in a real clinical situation.
It bridges that gap from the lab bench to the bedside, essentially.
It really does. Gives you confidence that this is something real and potentially very useful.
Okay, so if this microRNA is a reliable indicator, what does that actually mean for patients? For the millions with kidney disease? Or those at risk. You mentioned a dual promise earlier, diagnosis and treatment.
Yes, this is where it gets really exciting, the potential impact. First, as a biomarker, miR-423-5P could lead to a simple blood test.
Blood test, that would be huge.
Wouldn't it? Dr. Hebert pointed out, using this biomarker, a test could be developed to evaluate the status of the small blood vessels much earlier. Imagine that.
Instead of waiting for function to drop or needing a biopsy?
Exactly. A blood test could give an early warning about the health of those microvessels. Proactive assessment.
Which would be amazing for high-risk groups, right? Like the article mentions elderly patients or people having certain surgeries.
Absolutely. People undergoing organ transplants, major heart surgeries where blood flow gets temporarily stopped. Knowing beforehand if their kidney microvessels are vulnerable could completely change how they're managed.
You could put protective measures in place, monitor them more closely.
Right. Instead of just reacting after damage is done, it's proactive versus reactive.
Oh, okay. That's the diagnostic side. What about treatment? You said it might even be therapeutic.
This is the part that's really revolutionary, I think. Mignot found something incredible in the mice studies.
What was that?
He said that by injecting this microRNA into the mice with kidney injuries, they were able to actually preserve the small blood vessels and limit the kidney damage.
Wait, injecting it helped protect the vessels.
Yes. It's not just a warning signal. It seems to actively do something protective. It's a potential therapeutic agent.
That is incredible. So could you actually do that in people? Inject it?
Well, the good news is, during a transplant operation, direct injection into the kidney area is considered clinically feasible. The kidney is already exposed.
So you could potentially protect the remaining vessels right there during surgery.
That's the idea for that specific scenario. But obviously, you need ways to deliver it for other situations without surgery.
Right, for chronic conditions or prevention.
Exactly. So the CRCM scientists are already working on that, finding alternative techniques to transport the microRNA, maybe using tiny delivery vehicles or perhaps even a cocktail of different microRNAs to get it safely and effectively to the kidney.
Wow. So moving from just detecting damage to actively intervening to stop or reduce it, that's a massive shift.
It really is moving towards prevention and repair at a molecular level.
It just feels like such a significant step forward for kidney health. But it makes me wonder, could this go beyond the kidneys? You mentioned other conditions earlier. Is this microvascular damage a theme elsewhere in the body?
That's a really important question, and Dr. Hebert touched on this too. She noted that this loss of small blood vessels isn't unique to kidneys. It's linked to aging, both normal and accelerated aging.
So it's a broader issue.
It seems to be, yeah. It suggests that problems with these tiny vessels are a common thread in many chronic and age-related diseases.
Like what kind of diseases?
Well, the article mentions potential links to cardiac failure, pulmonary failure, or certain neurodegenerative diseases.
Heart failure, lung failure, brain diseases.
Oh.
Wow.
Think about it, in heart failure, poor microcirculation in the heart muscle makes things worse. In the brain, same thing. Impaired blood flow in tiny vessels can damage neurons.
It makes sense. It's all connected by this underlying plumbing, essentially.
Pretty much. And they even mentioned specific work by Emmanuel Bruchero, who's looking at pulmonary failure lung problems and investigating if microRNAs could protect lung microvessels, inspired by this kidney research.
So the insights are already spreading to other fields.
Yes, and there's another angle too. The CH has this amazing biobank, right, with lots of biological
The
samples.
one they used for the transplant patient study.
Exactly. They could use that resource to screen existing drugs, see if medications people are already taking, maybe for other reasons after a transplant, actually affect these small blood vessels.
So you could find out if a common drug is accidentally harming the microvessels, or maybe even helping them.
Precisely. It could help optimize treatments we already use, uncovering hidden side effects or benefits related to microvascular health. It broadens the impact even further.
So really, what began as a focused dive into kidney protection could ripple out and affect how we understand and treat a whole range of conditions. It's a fantastic example of how fundamental research pays off.
Absolutely. Understanding these basic biological mechanisms can unlock doors we didn't even know were there.
What an incredible journey today. This discovery about miR-423 5P, it feels like more than just another finding. It's a biomarker, yes, for early detection,
And a potential therapeutic agent, too. That dual role is key.
offering real, tangible hope for protecting those delicate kidney microvessels. It could mean earlier diagnosis, non-invasively, and maybe even proactive prevention or treatment for millions.
It really shifts the whole approach, doesn't it?
It feels like moving from firefighting late-stage disease to actually trying to fireproof the system much earlier.
That's a great way to put it. We're moving away from just reacting when symptoms finally show up, when a lot of damage might be irreversible.
Towards understanding and protecting these tiny, vital structures right from the start.
Exactly. Using molecular insights to potentially stop the disease process in its tracks, or at least slow it way down long before it becomes a major clinical problem. It's about getting ahead of the curve.
So here's something for you, our listeners, to think about. As we find more of these specific markers, like miR-423 5P, things that signal the silent damage, how might that change our whole view of health and aging?
Yeah, how could personalized medicine evolve? Could we one day routinely screen for the health of these delicate systems, microvessels in the kidneys, the heart, the brain, and intervene decades before symptoms even appear?
Preventing the silent decay before it starts. Powerful thoughts to ponder. Keep exploring, keep asking questions, and keep diving deep with us into the world of science.